In the 1980s, scientists recognized that certain regions in DNA consisted of repeating units of 2 to 6 base pairs, what we now call STRs. These regions were seen as highly variable between individuals. In 1991, Marian Litt and Jeffrey Luddy formally described STRs in a paper where they used the term microsatellites, which are similar to STRs. In the 1990s, STRs started to be used in forensic DNA profiling. STR analysis proved faster, more reliable, and required smaller DNA samples. Finally, in 1998, the FBI adopted a core set of 13 STR markers for its CODIS, Combined DNA Index System, database, making STRs as the standard in forensics DNA typing. The first time STR analysis was used in a court case occurred in the mid-1990s as STR technology began replacing older DNA profiling methods like RFLP, which stands for Restriction Fragment Length Polymorphism. One of the earliest documented uses of STR evidence in the U.S. court was in 1996 in the case of State versus Bogan in North Carolina. The case involved a sexual assault and STR profiling was used to match the DNA from the crime scene to the defendant. At that time, STR technology was still gaining recognition and courts were cautious. The Daubert standard, which was used to evaluate scientific evidence, played a big role in determining STR's admissibilities. STR analysis required much smaller samples, worked better with degraded DNA, and provided quicker results than older methods. After its successful introduction, STR profiling rapidly became the standard in forensics DNA evidence.
The short tandem repeat analysis is a widely used technique that remains a golden standard across a variety of different scientific fields due to its ability to distinguish individuals through their ideal DNA markers. One of the scientific fields includes forensics investigation. Here, experts use STR analysis to link victims, suspects, and distinct evidence to a crime scene. The STR profile of the individual can lead investigators to connect missing persons with unidentified human remains. Identifying paternity is another common field in which STR is used. The technique provides families with conclusive and defective answer to biological relationships they have among each other. Short tandem repeat analysis is conducted in medical setting as well. Its precise and sensitive analysis of individuals' genetic material allows professionals to monitor recipient and donor cells of bone marrow and organ transplants. This allows healthcare professionals to identify complications faster and track the progression of recovery. Overall, STR technique is a vital technique of DNA analysis and allows for new advancements in biology.
The steps for STR used for DNA profiling goes as follows. Number 1 would be to obtain biological samples like blood, hair, saliva, or anything that contains DNA. Number 2 would be to perform DNA extraction by isolating the DNA from the collected samples using methods to break down membranes and separate DNA from other cellular components. 3 would be to use PCR, polymerase chain reaction, to amplify the STR regions of interest in the DNA. This step makes multiple copies of the STR sequences to make sure there is enough DNA for analysis. Number 4 would be to separate the amplified STR fragments based on their size using gel electrophoresis or capillary electrophoresis. This allows visualization of the different STR alleles. Last, you would interpret the results to draw conclusions such as establishing identity or forensic analysis.
Requirements to make a match. To make a match, the sufficient number of loci, location of gene, must be analyzed. The more loci compared, the higher probability of a match. Number 2, there must be an eye-like consistency between the two DNA samples being compared. Number 3, statistical analysis is performed to determine the likelihood of the match occurring by chance. This involves calculating random match probability Number 4, the quality and quantity of DNA samples must be compared against databases of known profiles to determine if there is a match. And number 5, database comparison of the STR profiles must be compared against the databases of known profiles to determine if there is a match.